Good morning, everyone. I'm Simran Da Costa, and on behalf of Altum Technologies, I'd like to welcome you all to our new virtual webinar series on pharmacoinformatics. Today's session is entitled Exploring Potential Bioactive Components of Persea Americana for the Treatment of Rheumatoid Arthritis through Network Pharmacology. So, before we begin, I'd just like to give a very brief introduction to today's topic. I'm pretty sure that most of us in the life science and medical field may have heard of rheumatoid arthritis. So, this is a chronic autoimmune disease that is characterized by inflammation and joint destruction, and this leads to significant disabilities and reduced quality of life. Now, the current treatment options for rheumatoid arthritis have limitations, and in this talk, we are going to see network pharmacology methods to identify potential bioactive compounds from Persea Americana, more widely known as avocado, for the treatment of rheumatoid arthritis. We are going to see the databases used for collecting information on the phytoconstituents of avocado. We are also going to see the protein protein interaction network analysis, how key genes and proteins involved in rheumatoid arthritis were identified in protein drug interactions so that we can understand the underlying mechanisms of RA. So, let's understand more about this topic from our speaker of the day. It gives me immense pleasure to introduce Dr. S. Juby. Dr. Juby is an associate professor at the Department of Pharmaceutical Chemistry, JSS College of Pharmacy, UT. She has pursued a B Pharm in SB College of Pharmacy, M Pharm in KM College of Pharmacy, and PhD from JSS Academy of Higher Education and Research, Mysore. Her experience includes 22 years of both teaching and research. Her research areas are discovering novel chemical entities for cancer therapy through various in silico tools and drug scaffold repurposing. As part of the citizenship activities of the institution and the university, she was a member of the NAC and ACPE preparatory teams and is currently a member of the NBA IRF teams. She has published 87 international papers and 34 national papers in index journals with a cumulative impact factor of 155.02 in the range of zero point 0.4 to 13. Her total citations are 1,927 with an H index of 22. She has been granted 7 Indian patents. She has also published 5 books and 6 book chapters. Currently, she is carrying out 3 research grants sponsored by AICTE, RPS, ICMR, Government of India, and JSS Academy of Higher Education and Research, Mysuru. She has attended 19 international and 42 national conferences as a delegate. She has been invited as a resource person at 2 international and 21 national conferences and 19 webinars. She has guided 5 PhD students and 14 MPharm students, 15 BPharm projects, and 5 PhD students are currently working under her. She has organized many national conferences, webinars, and health awareness programs with the support of government funding agencies. So, without taking much time, I'd like to hand over the platform to our expert. So, welcome, ma'am. I invite you to take over the session.
Yeah, thank you, Miss Simran, for your nice introduction. Yeah, a very warm good morning to one and all gathered virtually. So, at the outset, I would like to thank the members from Altum Technologies Private Limited for inviting me and giving me this opportunity to share a little case study and the online tools which are explored in network pharmacology. Yeah. My topic is exploring potential bioactive components of Persea Americana, that is avocado fruit, for the treatment of rheumatoid arthritis through network pharmacology. So, in this presentation, I'll be taking you to various online tools where you can effectively use these online tools for your research. One case study is from this avocado. So before going to the study, the first 4 5 slides will be little bit introduction of the network pharmacology approach. So as we all know very clear, normally always we will depend on one drug, one target and one disease approach. Okay, so actually the drug discovery, all this computer aided drug design, it all started in the year of 1980. So before that, computers have been used only for the storage purpose. Okay, so the drug discovery, it starts from accidental discovery like penicillin's discovery, and or from plant source, or people will synthesize whether the synthesized compounds will give the activity or not. So these are the traditional approach. Okay, so the traditional approach, no, it has been replaced by the one drug, one target and one disease approach, that is the biomacromolecule. The scientists and the researchers, they are completely dependent on the target which is involved in the particular disease. Okay, though these one target, one drug, one disease approach, it has got a very good attraction, it also has got its pitfalls, that is like many safety, efficacy and sustainability challenges. So this is replaced by this network pharmacology. Okay, so here we will be playing with multiple targets. Okay, for some disease, as you all know very clear, cancer or some neuropharmacology diseases, Alzheimer's disease and all, it involved a complex pathway, and it involves many genes, many targets. Okay, so this network pharmacology approach, you know, it uses the computational power to systematically catalog the molecular interactions of a drug molecule in a living cell. Yeah, so this distinctive new approach, no, it is replacing the magic bullet based drug discovery to multi targeted drug discovery approach. We know very well the magic bullet based drug discovery approach, we particularly focus on a particular infectious organism. Okay, so your drug, it should act as a magic bullet, it should find out the particular organism and it should give its killing or sidal effect. Okay, so this network pharmacology and polypharmacology, they will go get along very well. Okay, so efficacy and toxicity problems, no, it will be taken care with this network pharmacology. Yeah, so this is a very good basic outline. Okay, so here derive some drugs for the treatment of obesity. That is my primary aim. Okay, so I should have compounds. So this compounds, no, it may be from synthetic origin or it may be from drug. Okay, drug repurposing, or it may be from the plant source. Okay, on the right hand side, you should have the target, which particular pathway, which particular molecular pathway we are going to work. Okay, so we have to finalize the genes and proteins which are involved in the particular disease, obesity. Then we have to construct the networks. Okay, there are two types of networks: protein interaction network and protein drug interaction network. Okay, so these are all some again online tools like gene ontology, a ket pathway, reactome pathway. Okay, so these, no, it will give you some insights about the genes and proteins which are involved in the particular disease. Okay, then you have to construct the network, compound target network. Okay, so like on the final, that is end of the target network, you will be having one or two compounds of priority which may work in the real scenario. So of course, all these in silico works have to be validated by in vitro and in vivo techniques. Okay. So the experimental validation goes, for example, for this obesity, we have to check the body weight loss, and some in vitro techniques like Western blot, flow cytometry, cell cycle analysis, Western blot, which particular enzyme is involved. We can estimate the particular protein, whether it is activated or deactivated. Okay, so this is a just outline of this network pharmacology. why this network pharmacology approaches, okay, why it is superior and compared to the existing in silico or other techniques. The first one is the comprehensive analysis. It will give you the full insight of the drug target interactions and simultaneously considering multiple targets and pathways, so leading to a more holistic understanding of the drug's effects and potential of target interactions. And the second advantage is rational drug discovery. Of course, here we use various computational algorithms. Okay, it may be online and offline tools. Okay, so again, it comes under the rational drug discovery. Of course, you can identify the novel targets which are not explored still. Then the fourth advantage is repurposing opportunities. The drug repurposing, because the pharmacokinetic profiling, no, the drugs already which are there in the market, so it can be repurposed for some other disease. So that is a very good option under this network pharmacology. And the fifth one is data driven prioritization. Okay, so this network pharmacology allows the prioritization of drug candidates based on their network properties, increasing the likelihood of selecting compounds with optimal pharmacological profiles for further experimental validation. So here the plant based network pharmacology comes, or it plays the major role, that is called as network ethnopharmacology. Okay, so during the progression period of network biology, natural products were gaining importance in the chemical space of drug discovery, as these have been economically designed and synthesized by nature for the benefit of evolution. So development of many databases, we have many databases which has the what history, complete history about these plant constituents or the constituents which are from the plant based or natural source. So, okay, so the network based approaches provide a systematic platform for the study of multi component traditional medicine and has applications for its beneficial modernization. Yeah. So with this brief introduction, directly we will be moving to the study. Just we are going to uncovering the potential active constituents of Persea Americana for the treatment of RA, that is rheumatoid arthritis. So the aim of the project is to identify potential therapeutic targets and natural compound. That is our ultimate aim. At the end of the study, we will be identifying the compound and we will be identifying the potential target, okay, which is used for the treatment of RA. So to achieve our objective, we use several online bioinformatics tools, online and offline tools, and analyze data on the phytoconstituents of Persea Americana. So it will be shortly called as PA, and this rheumatoid arthritis is RA. So coming to the disease, as given a very good introduction given by Simran. So this rheumatoid arthritis is a chronic autoimmune disease characterized by inflammation and joint destruction. Especially this disease affects hands, wrists, and knees. It will affect. Okay, and the rheumatoid arthritis signs are like as follows. It will be the bruised, heated and swollen joints, and usually it will become worse in the mornings, and there will be the stiffness of the joints, and normally these are the symptoms: fatigue, fever, and loss of appetite. Why we have chosen this Persea Americana? So this Persea Americana, actually this is a traditional medicine which is used for the treatment of inflammatory disorders in Central America. That is the first reason we have chosen. And this avocado, as we all very we know very clear that this is a greasy fruit, okay, and the delicious fruit it does, okay. It is mainly used for various culinary applications, okay, and especially the avocado oil, it is used in the cosmetic industry also, the present due to the presence of oleic acid. And majorly it contains the bioactive compounds for polyphenols, flavonoids, and fatty acids. And the third very important thing is it is generally considered safe for consumption, because safety is crucial when exploring potential treatments for chronic conditions like rheumatoid arthritis. Okay, what is the research gap and why we have chosen this disease? Okay, so the disease, it is highly prevalent throughout the world, this rheumatoid arthritis. And all, like what we are having the drugs, no, these are all used for the symptomatic treatment, especially the NSAIDs like COX inhibitors, ibuprofen or aspirin, or this disease modified anti-rheumatic drugs, DMARDs like methotrexate, okay, leflunomide. So these are all the symptomatic treatment only. So we don't have still an unmet need for more effective and safer therapies. Okay, and very another one is the pathophysiology is very very complex. Okay, the molecular pathway which is involved in the particular disease is very very complex. Okay, so we wanted to fill this gap. Okay, yeah, preliminary approach I can say. Okay, and contribute to the scientific understanding of the avocado's potential as therapeutic option for rheumatoid arthritis. Okay, as I told you, it is a complex disorder, this rheumatoid arthritis. Okay, so you can see there are so many signaling pathway which are involved in the RA. Okay, starting with notch signaling pathway, S okay, signaling pathway, PI3K signaling pathway, JAK, WNT, MAPK. Okay, so we all know very clear, during this inflammation, the pro-inflammatory cytokines like IL-2, IL-6, TNF alpha, so all will be evolved. Their level will be very, very high. Okay, a very good example is IL-6. Okay, interleukin 6. The interleukin 6 level is high. Immediately, it activates the Janus active kinase pathway and signal transducer and activation transcription factor, that is JAK-STAT pathway. So due to that, no, there will be a moment increase in the MMP, that is matrix metalloproteinase protein will be increased, and the what it leads to the arthritis, that is the synovial fluid accumulation, that inflammation it will happen. Likewise, PI3K pathway, it is involved in cell signaling. Okay, you have various signaling which is involved in this rheumatoid arthritis. So coming to our plan, so our plan, it has been divided into 5 phases. The first phase, of course, conducting literature survey. So the thorough literature survey on Persea Americana. Though the plan has been explored for the treatment of this inflammatory, this which particular constituent it is not fully explored, and we wanted to go with the fruits and leaves parts of the plant. Okay, so these are all we have collected from the literature survey. Okay, and the phytoconstituents have been taken from the very good database, impact database. Then afterwards we have gone for the in silico ADME prediction, the constituents which are safe to consume and which will have the safe pharmacokinetic properties. The second phase is very important, the target prediction for both for Persea Americana constituents as well as for the disease. Okay, and the third one is the third phase is identification of common genes, and the fourth phase is very very important thing, the construction of the network by using Cytoscape software, that is protein protein interactions and protein drug interactions. At last, the study end up with the structure-based drug design, the well-known molecular docking studies. Yeah. So this is the different tools what we have used. The first tool is impact. So this tool is particularly used for the collection of the plant constituents. Okay, the respective link has also been given. Then Open Babel, it is a online tool to convert the molecular format, which format we want for the study. Then Data Barrier, it is to check the ADMET properties. And binding DB, it will give you the genes. What are all the target genes from the Homo sapiens? Okay, and this gene net is disease associated database. It will give you the disease associated genes, and when it will give you the common genes which is involved in both, like plant as well as the disease. Of course, the cytoscape to construct network for both protein protein interaction and protein drug interaction. And Pyrex, it is an open online tool to perform molecular docking. And Discovery Studio visualization to find out to know the 2D and 3D interactions. This is the first phase of first and second phase, target prediction and gene collection. Okay, so on my left hand side you can see the plant name Persea Americana. So the corresponding constituents from the impact database has been collected. Then it has been undergone for ADME screening by using data barrier. Then by using binding DB tool, the genes which are associated with the constituents of the Persea Americana has been identified, targets of the PE. Okay, on my right hand side we have rheumatoid arthritis. What are all the genes which are associated with the particular disease has been collected. So we have both targets of PA and targets of RA. Then the network pharmacology, the networks have been constructed. So this is the first tool, Impact. It is nothing but very user friendly. It is nothing but this database which contains the Indian medicinal plants, phytochemistry and therapeutics. Okay. So, from the impact database, 253 phytoconstituents from Persea Americana have been collected. Out of these 253, 98 were the constituents from fruit, 155 were from leaf. Then, by using Open Babel software, the chemical structures were downloaded in MDL mole format. Subsequently, using Discovery Studio Visualizer, the 98 fruit constituent structures were converted into a single structure in SD-mol format, and 155 leaf constituent structures were also converted into a single structure in SD-mol format by using Open Babel software. Then the second tool is we encountered all these 253 phytocompounds into data barrier software. The software which will give you the in silico ADME properties, starting with its compound structure, molecular weight, molecular formula, log P, solubility, total polar surface area. Okay, and some toxicity also, mutagenicity, carcinogenicity. So all these things, no, it will give you the data barrier software. As I told you, these are all some important properties. So from this, the first filter has happened, and from this 253 it has been narrowed down to 64 active phytoconstituents. Okay, we all know very clear we have some limits, okay, for each and every property. The very good example is log P means it should be like around 5. Likewise, we have filtered all these 253 compounds. Okay, yeah, this is the table which depicts the properties, and these are all some constituents of Persea Americana, and these are all some important properties. Okay, and the some toxicity also, mutagenicity, tumorogenicity, all those things. So it is a continuation of the data barrier software table which gives you all the physicochemical and toxicity properties. These are the limits. Okay, based on the limits we have filtered. And coming to the step 3. So now all these 64 phytoconstituents, we have to predict the target. Each and every constituent is belonging to, or it is connected to how many targets. So for that, no, we used this binding DB database. Okay. So for the each and every compound, no, we have to collect the SMILES format, that is simplified molecular inline entry system. Okay, so that smiles notation of each constituent has been obtained from impact database, and it has been pasted in binding db. So here, very importantly, the constituents, only constituents with the similarity score greater than or equal to zero point 0.5 were considered. The zero point 0.5 has been kept as a limit. Whichever is more than zero point 0.5 were considered for these constituents. Okay. So it came to 25. Four of these constituents were from fruit, while the remaining 21 from the leaves. The target names of all 25 constituents were entered into the binding DB by the help of the UniProt database for the species Homo sapiens. We have to give the species Homo sapiens, okay, and the genes have been identified. Resulted in identification of 95 genes for fruit constituents and 404 genes for leaves constituent, a total number of 499 genes. Out of these 499 genes, some repetitions were there. So we have avoided all the repetitions, and finally it has been confined to a number 209. So these 209 genes are related or belonging to the fruit and leaf constituents of the leaves of the Persea Americana. It has been taken for the further analysis. Now, the Venn diagram. Okay, yeah. So before going to the Venn diagram, the DisGNet data has been explored. So this DisGNet has given the genes which are associated with the rheumatoid arthritis. The potential rheumatoid arthritis related protein targets were obtained from the DisGNet database using the search term RA, giving rheumatoid arthritis. Selected the summary of gene disease associations, resulting in retrieving 2723 target genes, okay, of rheumatoid arthritis. And finally, this is a very crucial step to identify the common genes which are associated in both rheumatoid arthritis as well as the Persea Americana. As I told you, this is the violet color. It indicates the total 2723 target genes of RA, and this it will give you the total number of 209 genes of the Persea Americana. So there are 86 genes, almost 3%, which is common to both 2723 as well as 209. Okay, so these 86 genes only we are going to take and we are going to use for our network construction. Okay, so these 86 targets represents potential targets for the active components of Persea Americana in the treatment of rheumatoid arthritis. The analysis of these targets can provide insightful thought or insightful what thoughts for the mechanism of action of these active components and their potential efficacy in treating RA. Yeah. Now, very important, the fourth phase is network construction. So, we got the target and we identified through this Venn diagram also the targets which are potential for PA and RA. So, these were entered into the Cytoscape network tool. So the Cytoscape network tool, it will give you both protein protein interaction and protein drug interaction. Yeah. So, in order to analyze protein interactions, the 86 drug disease intersection gene targets were analyzed. Okay, so the 86 drug genes, no, we are going to find out like how closely they are associating or how closely they are interacting with each other. That is this protein protein interaction network. Once the network has been constructed, okay, the it will give you the picture like this. Okay, so here the network consisted nodes. These are all the nodes. You can see here like SRC, MMP9, like matrix metalloprotease, EGFR, ESR, prostaglandin synthase 2. Likewise, the name of the gene, okay, it is called as node, and the edges are the connection between these two proteins. For example, the connection between AR and SRC. So this is considered as edges. Okay. So the network analysis revealed that the network consisted of 86 nodes and 527 edges, indicating a complex network of interactions between the targets of Persea Americana and rheumatoid arthritis related proteins. Yeah. So finally, we have to calculate, or we have to get these three parameters: degrees, betweenness, and closeness. Okay, so these three parameters will give you the how strongly or how closely the genes are associated with each other. Okay, degree is nothing but like how the two nodes are connected, or what you can say, how many edges can be formed. Okay, for if you take estrogen receptor, it can form how many edges. So that measurement is called as degree. Between us also, and closeness is how closely each protein is associated with another, or each node is associated with another node. Okay, so these three parameters are very 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 important. Okay, so based on these degree, this vascular endothelial growth factor A, it comes in the first place. Okay, and based on the betweenness, epidermal growth factor receptor EGRF comes, and based on the closeness, again this vascular endothelial growth factor A comes in the first place. Clear? Yeah. So the next step after the protein-protein interaction analysis was identifying the top plant constituents. So this was done by selecting both plant and target attributes in Cytoscape software, and using the Cyto NCA plugin to analyze the degree of centrality and between. Similar way, how the different genes or proteins are have been done, the network has been constructed. Similar way, here the network has been constructed for PDI also, that is the protein drug interactions. So these are all the different constituents like luteolin, alpha cardinal, like carva. I mean, so these are all the different constituents name. So finally, this is the integrated network of protein-protein drug interactions and PPI and PDI networking for both plant as well as the disease. And finally, the study has been ended up with the molecular docking. So after network construction, the potential, the top compounds have been taken based on the degreeness, closeness, and betweenness. Similar way, the top genes have been also selected. Then both know it has been undergone for the molecular docking studies by using online Pyrex software, and the binding energies have been calculated, and the 2D interactions also have been compared with the co-crystal of each and every protein. Yeah. So in this study, we identified the top 10 effective ligands and their targets in the treatment of rheumatoid arthritis in Persea Americana. First step, these are all well-known steps. Ligand preparation, it involves the preparation of the two-dimensional structures of ligands. Then protein preparation, of course, we have so many parameters, missing of residues and hydrogen atoms we have to add. So there are some standard protocol for the preparation of protein, and we have to find out the binding site amino acid residues, identification of active amino acid residues in protein by using uh, PLIP, protein ligand interaction profiler. Then the docking analysis, the next step involved molecular docking analysis. Yeah. So this is the sample table, starting with the prostaglandin endoperoxide synthase 2, and the protein PDB ID is 5F19, and the luteolin, the constituent of the Persea Americana, and the binding affinities, the binding energies in kilocalories per mole. It has been compared with the standard methotrexate as well as the co-crystal. Okay. Likewise, for all the proteins, the top five proteins like VEGFA, ESR1, SRC, EGFR. Okay. So all these things we have all the binding affinity calculations have been done. So among the five, the prostaglandin endoperoxide synthase 2, PTGS2, okay, it has got highest binding towards the luteolin phytoconstituent compared to standard methotrexate and co-crystal. Okay, of course, luteolin is showing maximum or very good binding score. Okay, to all the proteins. Among all the proteins, the prostaglandin endoperoxide synthase 2, it comes under a top scorer. Of course, we have to compare the two-dimensional interactions of our compound and with the standard as well as the co-crystals. Okay, so it is witnessing that compared to this, it has got a very good hydrogen bonding as well as van der Waals interactions and pi alkyl interactions also compared to these methotrexate and co-crystal. Yeah. So based on these studies, we have developed a new hypothesis. It is though the we know very well the avocado is well explored for the treatment of inflammation, but we wanted to identify among the genes associated with the rheumatoid arthritis, which comes first. So, based on these findings, PTGS2 is identified as a potential therapeutic target for rheumatoid arthritis. So, we all know very well the prostaglandins will be from arachidonic acid. No, arachidonic acid is the precursor. From that, the prostaglandins, no, it will be synthesized. So for the synthesis of that, no, we use this PTGS2, the prostaglandin synthase 2. So this prostaglandin synthase 2 will be inhibited by this luteolin, okay, a constituent of this avocado. So it will, what I can say, inhibit all these signaling pathways, the prostaglandin PGG2 and PGH2. So due to that, the inflammation is occurring. So these will be stopped. That is, the levels of expression of these proteins will be down. That is what our new hypothesis. So based on these findings, PTGS2 is identified as a potential therapeutic target for rheumatoid arthritis, and luteolin from Persea Americana, it is identified as a possible hit. Additionally, the PTGS2 gene was identified as a common gene between both Persea Americana and rheumatoid arthritis. PTGS2 was found to play a prominent role in the pathogenesis of complications associated with rheumatoid arthritis. Luteolin inhibits prostaglandin H2, that is PGH2 synthase, and therefore blocking the PTGS2 gene could potentially lower the inflammation of joints. So coming to the conclusion, secondly, this study focused on identifying potential bioactive compounds in Persea Americana for treating rheumatoid arthritis. The study's results identified several bioactive compounds, including luteolin, alpha cadinol, murolol, and others as potential candidates for the treatment of RA. Luteolin is present in both fruit and leaf parts of Persea Americana. And the PTGS2 gene was identified as a common gene between Persea Americana and RA. PTGS2 was found to play a prominent role in the pathogenesis of complications associated with RA, such as osteoporosis and cardiovascular disease. So, as I told you in the beginning, like our objective is to identify a potential target as well as to identify a potential compound. So PTGS2 is identified through in silico studies, and the luteolin is also identified. So as I told you, it is a preliminary study. With this, what can be explored further by using this network pharmacology analysis? What and all can be taken for further? So, of course, we have to do the molecular dynamic simulations to assess protein stability. That is the first thing. Suppose if we are very good in synthesis, we can of course develop some novel analogs derived from luteolin. We can study the structure activity relationship and we can go ahead further. Formulations aspect, the students from pharmaceutics background can go for formulations using this luteolin. And analysis, analyze and quantify avocado extract for cognitive and analysis purposes. Of course, finally the study should be about validated by using a proper in vitro and in vivo techniques using network pharmacology. And these are the references what we have used for the study for this network pharmacology study. And of course, I'll be ending my presentation. And finally, my sincere pranams to His Holiness Swamiji of Sutturmat, and my thanks to the entire leadership of JSS Academy of Higher Education Research, Mysuru, JSS College of Pharmacy, Ooty, the principal management of JSS College of Pharmacy, Ooty. Of course, my always my thanks to all my PG and PhD students. And this particular work has been carried out by a group of BPharm people, uh, like Mr. Chandru, Miss Saubarnika, Mr. Sudarshan, and Mr. Dhanush, and guided by my scholar, Mr. Ake Krishnaswaru. Special thanks to all of them. And this is my profile page, and I have authored a textbook which is for UG final year students, especially for B Farm and first time form students. And basically, my research work is 30 to 40 percentage of in silico, followed by synthesis, then finally in vitro and in vivo validation. So during the COVID time, that is cata, that is one of the plant, especially mm, 3,4 dihydroxyphenyl trihydroxybenzoate. It is used for this catechin, like what I can say, the green tea polyphenol. It is mainly used for SARS-CoV-2 target. So that paper has come as hot articles in RSC Advances. So with this, once again, my special thanks to Altum Technologies for being inviting me and sharing my just whatever I learned in this network pharmacology. Special thanks to Miss Divya ma'am and Sakshi, and of course my special thanks to Miss Sheetal and Simran. So thank you all. I am very happy to answer if you have any queries. Thank you.
So, thank you, Dr. Julie. This was a very knowledgeable session, and thank you for having a very clear presentation and including a future perspective slides, because we have a lot of students watching this as well. So, that would help them get a very good clarity on how to proceed further in a related field. So, it's time to take some questions now.
Yeah,
Do we
sure,
have any
sure.
questions? So the first question is how network pharmacology works on autoimmune mechanism of rheumatoid arthritis.
Yeah. So I think you can go for this Jack stat pathway also, or other. You have to find out which particular cytokine is majorly involved. Of course, it is involved, like so many cytokines are involved in that autoimmune mechanism. So accordingly, you can select. So I think one of my student has been extensively worked in IL-6. I think you can work on this IL-6 also.
Thank you. The next question is how do we know that luteolin is not toxic?
Yeah, it's a very good question. The previous during my presentation itself, the starting of the presentation itself, I told it is an in silico prediction. Of course, you have to do the toxicity studies, okay, by using the in vitro cell lines, or you have to go with acute oral toxicity studies, okay, before going to the preclinical studies. Yeah, definitely you have to check the toxicity. In silico level, it is very safe to consume, but of course, you have to validate experimentally.
Can we have the next question, please? The next question is, how do you think the antioxidant properties of avocado can help in managing RA?
Yeah, this polyphenols, no, especially this luteolin and other polyphenols are having very good very good antioxidant properties. Okay, so but the mechanism I am not very much sure. We have checked only the inhibition of the top five genes, okay, and especially the PTGS2. We have to study extensively again for this antioxidant property. We have to connect antioxidant genes and this luteolin or other flavonoids. Yeah.
The next question is, any tissue specific study for RA performed to identify drug targets?
Specific study, I think for osteoarthritis, we have a study that Flynn adjuvant model, okay, for animal studies. For rheumatoid arthritis also, I think there may be studies available. I think you can have a look on it.
Thank you.
The betweenness, how to find betweenness? Yeah, it is again a mathematical number. The There is one plugin is there, cyto hub plugin is there, okay. So the moment we give that option, no, it will give you the score, the betweenness. So the betweenness is how closely the two nodes are associated. Again, it is the number. You can see my presentation. 1 minute, I'll show the presentation. It is there. Yeah, this is the betweenness, the number. So the higher the number, the betweenness, the association is very strong. So in this case, we can tell the luteolin has got the higher score.
Thank you, ma'am. The next question is, could you tell more on network pharmacology approach for potential bioactive compounds identification for other disease types?
Yeah, definitely you can. That's what I told. You can go ahead with any disease, especially for cancer, Alzheimer's disease, okay. It involves the complex pathogenesis. So such type of diseases you can go ahead very well. I suggest cancer, you can go ahead. This osteoarthritis also you can go ahead. Then for Alzheimer's also you can go.
Thank you. Any more questions from our participants? Okay, the next question is, as I know to calculate top 10 degree, but I wanted to know how to find out betweenness.
Yeah, you can. I think after this presentations, you can reach me. I will teach you how to do it.
The next question is, how do we know that the compound identified will not have any off-target interactions with other human proteins?
Yeah, this is also again very good question. So all we all know very clear, this all these in silico thing is all it is relativeness, okay. Always we will compare, okay, and we will take the top prior compounds only, we will take it, okay. So definitely maybe apart from PTGS2, the compound will have the interaction with other protein also, but our prime aim is to identify the top compound, top one or two protein. So this question we have to answer with once we do the experimental validation. Yeah, of course, we have to do the enzyme binding assay with PTGS2 or gene expression studies with PTGS2, then we will come to know. Suppose if we have got any negative results, then the second or third compound will have.
Thank you. So it looks like we are done with our questions.
Yeah, thank you all for your comments. Yeah,
So thank you so much, ma'am, once again for giving your valuable time and taking this session. It was very
yeah.
informative.
Thank you, Simran and Sheetal. Yeah, thank you. Shall I leave?
Yes, ma'am. Thank you.